Hello. Hi, everyone. Good morning. Thanks, Dr. Sedel, for the introduction. My name is Karteek. I'm an assistant professor at the Department of Computer Science, and I run the Data and Image Analysis Group Lab, the DIAG Lab. And today I'm going to kick off this wonderful meeting with a talk on my work that one of my focuses, which is developing biomarkers, imaging biomarkers for dementia diseases like Alzheimer's. So I'll first give you a brief overview of what I do in my lab. So in my lab, this is what I look at day in and day out, me and my grad students. So we look at medical images, and our goal is to develop technologies, basically computer software that can automatically interpret these images. So just to set up the context for the problem, so currently, if you go get any sort of medical test done, like a physical test, like if they take a blood sample or some other fluid sample from your body, we don't directly take the fluid sample to the doctor and say, hey, please tell me what's right or wrong, right? So we go to like a diagnostic lab, which will analyze the sample for various biomolecules, and they generate a report, and then the report is given to the sort of like the doctor, right? Because the report would make much more sense than doctors directly looking at the samples. But when it comes to images, images are also like samples. It's just that they're digital samples of your body, right? But what happens with images is we just directly give them to doctors called radiologists, and they literally visually look at it and do the equivalence of what these diagnostic labs do, but in just with their physical senses, right? And you can see it's a very hard problem, right? So I'm just giving you a sort of like a smattering of images here. So this is a MRI image, which is highlighting the brain tumor here, and there is an image showing the glucose activity in the brain, and this is the image of a body, whole body CT image. These are the lungs, and these are the legs, and these are images of retina, vessels. So there's so many images, and doctors have to look at these and come up with sort of answer the question related to patient health just by visually looking, right? And this is the problem I'm trying to solve. Uh, to further motivate the problem, the images that you see are not 2D, but they're actually 3D. So we actually put a patient like this in some sort of like a scanner, and they're scanned from like head to toe, and the images are really 3D in nature. So I'm just going to scroll through a couple of images you see. So a 3D image is basically a collection of 2D slices. So you can imagine how challenging the task is for a human to just look through these and find out the patterns of disease, right? And this is again, this is simulating sort of like the traditional it's called a light box image. In the olden days, they used to print them on the sheet of paper, plastic, and then look up to the light box and look at the images, right? So ironically, that's really what doctors are using computers for when it comes to images. Like radiologists, they're using a computer, but they're not really using the sort of any technology from the computer to help them interpret the images. They're literally using them as viewing devices, right? For the most part. So my goal, my lab's goal, is to do something like this to the images, right? So develop, automatic algorithms that would color the images so that it makes it easy for the doctor to, focus their attention, right? So in this huge minefield of imaging data, pool of imaging data, you focus on, the structure or the pathology of interest, and better, once we get those, we can extract measurements and we can make the reports I was talking to you about, right? So you can just say, oh, here, these are the various, volumes of these structures, or, texture characteristics of a structure, which could be indicative of health and disease, right? And then we can also make like cool visualizations, population level. So, and also some 3D visualizations, which enable, and this could be beneficial both for the doctor and for the patient as well, to see how their, internal body health is, right? And ultimately, so we might say, hey, you have now transformed a problem from big imaging data to big numbers now, right? There's so many numbers now. How do I deal with this? So this is where, again, we can use machine learning and condense all these numbers into like maybe like a health score, right? Just like how you have, now we know like the ideal blood pressure is 120 by 80, right? And they give a normative range. Like that, we can give, for instance, a measure of 0 to 1 for brain health, 0 being healthy, 1 being unhealthy, and then someone could lie anywhere on the spectrum, right? So there are really like three boxes, and my lab works on developing technologies for all the three boxes. So really the holy grail of what I want to do in the end, or the grand vision for my research is, in goes an image, not just an image, multiple images from all these various cameras, so-called scanners, MRI, CT, PET, and from the various parts of the body, and then they're all condensed into like a unified health score, right? So that's the idea. So today I'm just going to show you some slides on the specific work I've done on coming up with such a number for brain health, and with a focus on Alzheimer's disease. So basically, we'll come up with a number between 0 to 1, where 0 means the patient is clear of no signs of Alzheimer's, to 1 is they have Alzheimer's, and anywhere in the spectrum we'll say like is like a risk of Alzheimer's, right? So here's just a snapshot to show you, to motivate the serious problem with Alzheimer's disease. So Alzheimer's disease is a disease that basically eats your brain, right? And the thing is, the disease starts way before the symptoms occur, right? And by the time people see symptoms, unfortunately, the patient's brain looks like the one on the right side, right? It's irreversible. So because the brain tissue is dead, there's nothing you can do, right? So the challenge in Alzheimer's is really to catch the disease as early as possible, right? I mean, even though currently there are no treatments exist to reverse Alzheimer's disease, but there's a lot of research going on to develop those, right? And we hope that at some point we would develop such treatments. But for such treatments to be effective, we still need to catch the disease early enough, because there's nothing you can do once you catch the disease at an advanced stage, right? And so one of the tools we can use is the what are called FDG PET images, right? So these are basically, like I showed in the first slide, these are images of the brain where a radioactive tracer is injected, and then the tracer attaches to glucose molecules. And it has been observed that neurons that are lazy, or like which are start to stop functioning properly, will uptake less glucose, right? And in a sense, they will not light up on the image. So neurons that are healthy will light up because they take up the radio tracer, and then we detect the radioactivity from the tracer. So if in this scan, if you see the red areas are where the uptake, so the redder, hotter colors are where the neurons are healthy, and the bluer, lighter colors are where the neurons are unhealthy, right? So now we get a 3D sort of map of the brain, right? And the idea is, using this 3D map, can we convert that into a single sort of 0 to 1 number, right? So that was the goal of this project, right? And for this, we also use MRI images, because FDG PET doesn't tell you the structure, right? It only tells you, it only tells you the function of the brain. But we also wanted to overlay it with the MRI images and see which organs, which substructures in the brain are, uptaking how much, and use those as features. So this is what the text is saying here. We identify like 85 regions in the brain, and we computed like average signal in each of those uh, uh, brain regions, and then use that as a sort of like a biomarker, like initial biomarker for the patient, and try to build a machine learning model to convert that into a 0 to 1 score, right? And we also did a modeling of the disease itself. So as you can see, so one of the highlights of the study was we did it on a large set of images, around 3,000 images taken from 1,300 patients, right? And we also modeled Alzheimer's not just as a disease of 0 or 1, Alzheimer's or not Alzheimer's, but as sort of like a spectrum on a longitudinal scale, where people at this spectrum are completely healthy, and people at the end of the spectrum are confirmed Alzheimer's, and people in the middle are called mild cognitive impairment. These are the people showing some signs of impairment, but we don't know if they have Alzheimer's, right? And this is actually the real meat of the problem, is to identify which of these people will go on to get Alzheimer's, right? So it's kind of like saying, is my grandfather just getting elder and he's just natural aging, or is it displaying signs of advanced aging owing to disease, right? So uh, so what we did was we said we will model the problem by taking the extreme ends, taking the signatures, FDG PET signature of the extreme ends, and build a model, and then predict everyone in the middle and see if that model is able to put them in the right order, meaning people closer to the normal timeline will get lower scores, and people closer to the what we call the DAT plus, dementia of Alzheimer's type positive side, will get higher score, right? And this is a visualization. So before we started the, obviously building our machine learning models, we just try to visualize all the patients in the database. So the x-axis represents the patient. So each point on the x-axis represents a patient, and each point on the y-axis represents all the 85 regions that I was talking to you about. And the color basically indicates the deviation of the average FDG PET signal in that patient in that region from the normal population, normal average, right? So wherever you see blue, that is actually bad, and red is good, right? So as you can see, and these are laid out in from the normal to Alzheimer's spectrum. So you'll see as you go from left to right, you'll see the number of blues increasing, right? So this gave us a sort of like an indicator that the base feature that we have chosen is kind of in the right direction. So it might really help us identify this number, right? Alzheimer's number, which we call the FPDS, FDG PET dementia of Alzheimer's type score. And then this is, the slide just showing the machine inside the black box of the machine learning model that we trained. As I said, we trained on the extreme ends, and we tried to classify people in the middle. And this is the sort of like the results we got. So the final, so like I said, we call the biomarker the FPDS score. And so let me just highlight you the key number here, so which is called the AUC. So basically, AUC measures how well the algorithm was able to separate the two sort of groups that are tending towards Alzheimer's and groups that are not tending towards it. And again, AUC of 1 means the algorithm was perfectly capable of separating everyone with 100% accuracy, which means it didn't predict people from here, this group to that group, or vice versa. And 0 means it failed completely, right? As you can see, we got a pretty good accuracy of close to 78%, right? Which is, again, this is up for debate. Is this clinically useful yet, right? So one of the things with machine learning algorithms is we have to do a lot of independent validation, right? So the proof is in the pudding. Like when you do your little research study, so obviously this number, we derived it by keeping aside a bunch of images as completely unseen, like we said, right? We only used images from the extreme groups to train, and the middle groups were not used to train. So they were kind of independent. But I would still claim that that is not independent enough. The real test is to give this to clinics around the world, and they test on real patients, right? On completely unseen data, unseen images taken from different scanners. And there's all this variability. And then if the numbers still hold, then we can claim that we are closer to, and that's one of the pushes I do in my research as well, is to make these tools available as an easy to use software. So there's a big push on engineering that as well, because I feel like these tools just developed in the lab, most often just stay in the lab, right? And we never know their true potential unless we deploy them. So yeah, so I think that's basically the my talk for today, and I'd be happy to take questions at the end of the session. Thank you. And I would like to introduce next speaker, Dr. Sarah Paul.